Now we'll move on to our support pool fund speakers. I think Shirley Sunoto is going to be introducing the support pool funding award from UCSD.
Thanks, Sarah. Good morning, everybody. I just wanted to thank and acknowledge the NICHD for the support pool funding. This year, I believe it was Prince yesterday who mentioned that because of the timing of everything, most of the centers utilize the support pool funds internally, which is what we did this year. But I just wanted to put a plug out there that we're very happy and willing to collaborate with any of you in this next go around of the support pool and opportunity pool funding, which I believe the letters of intent are due July 1st. So this project was spearheaded by Sydney Thomas, and there's multiple people collaborating on it. And I hope this again showcases sort of the power of untargeted metabolomics and in interrogating breast milk. So she did a multi-omic analysis of human breast milk to identify xenobiotics and other chemicals. And Christina Chambers and Lars Bodie were also involved in this project. Next slide. So we had two aims, and Sydney's going to be talking about this first aim, which was where we conducted a multi-omic analysis of existing milk samples that were recently treated with antibiotics. And we also have a second aim in collaboration with the Vanderbilt site, where we're helping to also utilize genomic information and add this to our multi-omic analysis. And right now they are currently attempting to extract DNA from existing human milk samples. And if that's successful, we will be sending them some samples that we have so that we can pair some of this genomic data with the other multi-omic analyses that Sydney's going to be conducting. So Sydney, please.
Hello. Next slide. Thank you so much, Shirley. So my name is Sydney Thomas. I'm a postdoc in the Dornstein Lab, and I'm going to give a brief update on our multi-omics analysis of human milk. Next slide. So this particular analysis looked at 550 human milk samples where we had data on 16S microbiome sequencing of these samples, untargeted milk metabolomics, as well as concentrations of several different human milk oligosaccharides, or HMOs. Next slide. So one of the first things we did is look at the diversity of each of these three data types under several parental medical conditions or medication uses. This is way too small to see and way too early to look at. So we're just going to focus on a couple of categories here. So if you click forward, we can see that we found this interesting phenotype where under certain conditions, you see a decrease in microbiome diversity that is accompanied by an increase in HMO diversity. And so we were interested in whether there are any specific human milk oligosaccharides that were responsible for this effect. So on the next slide, you can see that we mapped each of these different human milk oligosaccharides that we measure to microbiome diversity. As you can see, many different HMOs are associated with either positive or negative microbiome diversity. What was really interesting to us, though, was that there were three specific HMOs, 6SL, DSLNH, and LSTC, that were both negatively correlated to microbiome diversity and significantly upregulated under certain conditions, such as preterm birth. And so when we showed this data to our collaborators, who are imprint and who are HMO experts, they told us, well, it's obvious why all three of these HMOs have the same phenotype. They actually all share a similar structure and are created by a single enzyme, ST6-gal1. And so with that, we started to look at the sum of these three HMOs together. And what we found is that if you take parents whose milk has concentrations of these three HMOs in the upper quantile versus the lower quantile on the next slide, you can see that parents on the upper quantile were twice as likely to have had a C-section, twice as likely to use SSRIs, and 5 times more likely to have had a preterm birth. And so this was a really interesting phenotype, and we wanted to know if there are any other changes in milk composition that were associated with these three HMOs. And so one of the first places we looked was in the milk microbiome. So that's on the next slide here. This was done in collaboration with another great postdoc in our lab, Dr. Simone Zuffa. And what we found was that while there are many bacterial genera that are associated with maybe one or two different of these HMOs, which are shown in the triangles here, there are a group of bacterial genera that are associated with all three HMOs and all in the same direction. So that really indicates that these HMOs are changing the microbiome of milk in some really interesting ways. And this HMO phenotype isn't just associated with changes in the milk microbiome, it's also associated with changes in the milk metabolome. So on the next slide, thanks. On the left, you see what's called a molecular network. This is a tool we use in untargeted metabolomics to group molecules by structural similarity. So we don't actually know the exact chemical identity of any of these particular molecules, but we do know they're peptides. We do know they're structurally related to each other. We're working on annotating them. But you can see that almost all of them are significantly associated to concentrations of the three HMOs of interest. This doesn't just happen for peptides. On the next slide, you can see that we have other classes of molecules that seem to be significantly associated with these three HMOs. I'm showing phosphatidylcholines here, but we also have some other lipid classes that I don't have time to show you here. So that really indicates that these three HMOs, we see changes in the microbiome, we see changes in the metabolome, we're seeing these kind of big changes in milk composition. And so finally, we wanted to see whether those changes in milk composition actually affected infant health. So to do that, we mined EHR data from child outcomes up to 36 months paired with these parents. And we found that children of parents in the upper quantile of these HMO concentrations had significantly higher scores on externalization at 12 months and internalization at 30 months on the infant toddler social emotional assessment. Here, higher scores are worse, so that indicates that these children have deficits in both externalization and internalization of emotions. We also saw that these children had significantly less percent phrases understood at 24 months based on the MacArthur-Bates Communicative Development Index. And even at 36 months in panel C here, you can still see a significant correlation between dysregulation of negative emotions based on that ITSE test and concentrations of these three HMOs in the parent's milk. So in conclusion, next slide, what we've found so far is that milk composition may be predictive of preterm birth and infant development. Integration of multi-omics data has been giving us some really interesting new insights in how this milk composition affects parent infant outcomes. And next, with future work, we're focusing on replicating these results in an independent cohort and understanding the biochemical mechanism behind this phenotype so that we can find hopefully new biomarkers or specific targeted interventions to improve both parent and infant health. So lastly, thank you to everyone who is involved in this project. It really takes a village to collect and analyze all of this data. And I'd be happy to take any questions after all of these talks are done. Thank you.
Thank you, Sydney. Now we'll turn over to